Good evening, everybody. Welcome to the research workshop for the second year of the High School Science Symposium of Robert College. We want to extend our thanks to Evrimacı and Evrimacı Lise Forum for their contribution in the organization of the workshop. And we want to thank Dr. Semih Taren for accepting our invitation and giving this workshop on key principles of performing academic research and writing an academic paper. We will start with Dr. Semih Taren giving his workshop, then we will proceed with Q&A if you have any questions. Afterwards, our HiSci team members will talk more about the research template for HiSci 2022. And Dr. Semih Good evening, everybody. Welcome to the research workshop for the second year of the High School Science Symposium of Robert College. Sorry for that inconvenience. Dr. Semih Taren earned his bachelor's degree in microbiology from the University of Washington in 1998. He received his PhD degree in molecular and cellular biology from the University of Washington in 2010. Currently, he is the senior director of Sana Biotechnology. His field is virology, and his expertise in viral vectors and gene therapies. Thank you again for joining us, Dr. Taren.
Thanks, everyone, for having me. I appreciate the invitation. So this is my first time talking to the Robert College, I believe, and the good friends over there asked that I come and present and I talk about how research is done. Is that correct? Excellent. All right, so first of all, one thing you'll notice about my talks is my talks are a little unconventional. And that's because I like to do research in a bit of an unconventional way. I like to live a bit of an unconventional life as a scientist. I have many interests that I want to talk about because the main purpose of my conversation today is to be able to hopefully inspire the next generation of scientists like yourselves. So I'm going to do a few presentations. I'm going to go in and out of slides and try a few presentations as well, just to get everyone oriented, and we'll go from there. So if you have any questions, please feel free to stop me. All right, so I'm going to first tell you a little bit about myself, and that's going to be important because it will allow you to get an idea of who I am as a scientist and the type of different people you can meet in the science field. So I am a scientist, yes, but I also spend a big chunk of my time as a composer. I compose music for horror films. And whenever you think of a scientist, when you Google the word scientist, you come across a lot of images that look like this. And sometimes these are characterized by old white men with white hair and glasses who are doing crazy and often silly experiments. And in the United States, maybe not so much in Turkey, but in the United States, Bill Nye, the science guy, pretty much popularized the popular image of what a scientist is. And all these are great inspirations, but I want to change the norm a little bit. I want to show you this image. This is a band called Insomnium. This is one of my favorite music bands that I enjoy listening to. They're from Finland. And one of the reasons I'm sharing them is because one of their musicians is Dr. Will Freeman. He is a professor at University of York, and he lectures in evolutionary biology. And there's his photo. He studies microbial protists of prophages, and he works on living fossil records and the evolution of life. Another example I want to give is this lady here. She's a wrestler, and she's won many awards, and she's also a scientist, and she goes by the name Rachel Collins over here. All right, so, and then this is me. So I live a life where during the day, I'm a scientist in the lab, and I shouldn't say during the day, but day and night, I'm a scientist in the lab. I work in biotech. I work at Sana Biotechnology, but then I also have a life where I'm a composer, and I compose music for horror films, and I've been doing this for the last 20 plus years or so. And so today our topic isn't film music, but I wanted to open this way because I wanted to highlight that in order to follow whatever your passion is in life, whether it's science, whether it's music, whether it's art or literature, mathematics, engineering, law, that you Make sure that you bring your own identity to the type of work you do. So I think most of you may know my story a little bit, but I'm originally from Izmir. And then in 1995, I migrated to the United States of America to Seattle, Washington. So Seattle, Washington is in the northwest corner of the United States, very near Canada. And right now I'm calling you from Seattle, Washington, and we have about 11 hours of difference in time. So in Turkey right now, it's 8 p.m. In Seattle, it's 9 a.m. Saturday. Thank you for introducing me in the beginning with my education. Just to remind folks, so I started my science education at Ege University in Izmir, studying biology, and then I transferred over to the University of Washington. University of Washington, I got my bachelor's degree in microbiology, and then I worked for many years as a research associate. And then I got my PhD at the University of Washington and at the Fred Hutchinson Cancer Research Center. My PhD was on the topic of virus evolution, specifically studying viruses related to HIV, so retroviruses. And then as a career, I chose to be in industry. So in science, as a career, you can choose to become an academician. You can choose to be in industry like I am. You can choose to work for the government. You can choose to be a consultant and be self-employed. Or you can choose many other aspects. I have many scientist friends who got their PhD. Some of them decided to go and study politics. So they're working on science policy at the White House. Some of my friends chose to become lawyers. So after their PhD, they got their law degree and they're working as patent attorneys for science companies and so on. So there are many, many ways to do this. You don't have to assume there's only one way. Often when we're in school, we're taught that the only way to become a scientist is to get your PhD and then become a professor at a university, but that's not true. So I wanted to join the industry because I wanted to work on applied research that directly resulted in therapies to cure diseases. So I started working at a company called Eli Lilly, and Lilly is a giant pharmaceutical based in Indianapolis, Indiana. Lilly is famous for the first diabetes, recombinant diabetes medicine. They're famous for Prozac and so on. And then I worked at a company in Seattle called Cell Therapeutics, and then Immune Design, Juno Therapeutics, and Sana Biotechnology. So for the last 3 years, I've been at Sana Biotechnology. And I'll tell you a little bit about what I do. I'm going to skip this part real quick because our topic is not music today. All right, so here's what I'm doing right now. So my PhD thesis was titled this: Molecular Consequences of Virus-Host Interactions as Inferred by the Function and Evolution of Host Antiviral Protein TRIM5 alpha. It sounds like a mouthful, but often PhD theses can be this way. And I right now work on developing viral vectors to cure diseases like cancer. So my team and I, we use viruses to train the immune system to attack cancer. And here's a cartoon of what that looks like. So this is a specific kind of therapy called CAR T cell therapy. And we bring in the sick patient, so the patient who has cancer. We apherese them, so we isolate their blood cells. We use viral vectors to transduce their blood cells. So these are viruses that are derived from HIV. They're safe, they won't cause disease, but they also carry a very special gene inside of them. So with this virus vector, the gene is delivered to the white blood cells, the T cells of the patient. This happens in the lab. This gene allows the expression of a very special protein called a CAR, or a chimeric antigen receptor. This special protein teaches these cells to recognize and kill tumor cells. You grow these cells in the lab until they reach a certain number, and then once your infusion is ready, you call the patient back in, and the patient comes in and you infuse them their cells that are now engineered to recognize and to kill cancer. And my team and I focus on these two areas because we are a team of virologists and molecular biologists where we engineer and overexpress these CAR molecules within the viral vectors and make sure that they do what they're supposed to do. And in 2014, we started working on a therapy that finally got approved last year in February. It was approved by the FDA. So the FDA, as you may know, in the United States is the organization called the Food and Drug Administration. They authorize medical products, they authorize vaccines, therapies, and so on. And so here are the headlines from February 5th with the approval of our therapy. And so now we're working on a few different therapies. And instead of this process that I just mentioned to you can be a bit cumbersome because it takes 2 to 3 weeks. And sometimes these cells in the lab may not work really well. So we're trying to come up with a simpler process right now. Instead of bringing in the patient, apheresing them, transducing their cells in the lab, growing their cells in the lab, and so on, we are trying to design viral vectors that would allow us to eliminate all of that and instead simply allow us to directly deliver the viral vector to the patient. Just like HIV would infect T cells, here we would have a viral vector that would target T cells, it would be safe, and it would still allow the T cells in the body to be trained against cancer. And one of the ways we're doing this is by changing the outside of the viral vector. So here's what I mean by that. We have a viral vector here on the right that looks like this, but on the outside, we are adding molecules, specifically fragments of antibodies that allow this viral vector to target CD8 positive T cells. So in the blood, the immune system comprises of many cell types. These can be T cells, B cells, monocytes, CD4s, CD8s, etc. And using this technology, by adding antibody fragments, you can target any of these cells that you want. So let's say we chose to target CD8 cells. So this viral vector would target CD8s and only CD8s and not the other cell types. And then once you transduce this into the patient, hopefully it should be able to target these cells and grow them. All right, and another thing I wanted to get to is the topic of today. So I'm going to switch my presentation to a different topic here. And I want to talk to you a little bit about how research is done. So since that's the topic of today. All right, so here we go. So I'm sharing with you now a diagram. This is a diagram of a cell. So this big blue circle is a cell. And here is a lentivirus. A lentivirus is a type of family of viruses. HIV is a member. And we use lentiviral vectors. So the viral vectors we use to transduce cells and deliver genetic information is made from HIV. And our therapies are all dependent on being able to deliver the genetic information to the cell, because with this genetic information, we are able to teach these immune cells how to attack and kill tumors. Now, this diagram is showing how difficult that journey is. When you design a viral vector, and when you want that viral vector to infect, or we call it transduce, when you want it to transduce any cell type that you want it to be, that virus has to overcome several barriers. First of all, it has to recognize the receptor. It has to be able to enter the cell and transmit through the cytoplasm. It has to overcome several biological immune factors. So inside the cell, there are many immune factors that protect the cell from the virus. You may have heard of antibodies. We hear of antibodies all the time. Today, during COVID-19, we hear of antibodies all the time. We hear of cellular immunity all the time, but there's also a third kind of immunity that happens inside the cell, and we rarely talk about this. The reason is it's a new field, and the majority of public are not familiar with this, and also the majority of news outlets aren't familiar with it either. But inside of your cell, there are immune molecules that recognize parts of the virus. And mind you, at this point, the virus isn't a whole virion. It is parts of the virus, such as the capsid, the nucleocapsid, the matrix, the RNA genome. It's these molecular parts of the virus that these immune factors recognize. Some of the names of these immune factors are shown in this diagram: SAMHD1, ApoBec3. These recognize parts of the virus and they block its ability to complete its life cycle. And similarly, when a virus is being made from a fresh cell, some of these restriction factors are able to block the packaging of the virion and the release of the virion. One of the famous factors that blocks this is called tethering. These are all host molecules in our cells. And when we design viral vectors, we have to be able to overcome these. So I'm going to tell you a short story about how we went about doing this. So we were developing, let me pause the slide for a second. We were developing viral vectors that we could inject into people in their skin. And these viral vectors came from HIV, but on the outside, they had an envelope that came from Synvis virus. Synvis virus is a mosquito virus, and we were using a mosquito virus on the outside so that we could take advantage of infecting at the skin. We were trying to infect dendritic cells. Dendritic cells are a type of immune cell that are present in the skin, but there was a problem. These dendritic cells had a restriction factor similar to the one I told you about. And we had to figure out a way to overcome this restriction factor. So now I'm going to share the work that we did just to give you an idea of how we went about doing that. So this was the publication that we released. This is PubMed. So PubMed is a website where you may be familiar with. You can, it comes from the NIH, and it allows you to search for publications. So this was our publication that described what we had to do to overcome this restriction in these dendritic cells. And if you're not familiar with these websites, you can search by author or project or name. And once you search them, you'll get different hits. And these hits will tell you the name of the journal. So this is molecular therapy, the date, and then the DOI numbers, and so on. The title and the authors, and then usually you'll have an abstract. And also on the same page, you will see if there are any full links, you'll see where they are. So let's go ahead and open one of these full links. So I'll give you an idea of how this research was done. This is now the journals page, Molecular Therapy. And in this page, you will see some of this work that was done. This isn't a very good format. Let me switch to the PDF, pardon me here. There we go. This is the PDF. This is a much easier format to follow. So I'm skipping to the first figures, and the first thing we had to make was we wanted to make a viral vector that didn't integrate, because this was supposed to be a tumor vaccine. And normally these viral vectors integrate. So we had to design a viral vector that wasn't going to integrate. And that's what we did first. So first we made an integration deficient viral vector. And that required us to add two components, to modify two components of this vector. And that's what these two experiments are showing here. Looking at the percent of integration on the y-axis. Here is wild type and the amount of integration. And then we made these changes shown on the right here with these numbers, D64V and 704. And this abolished the integration by 3 to 4 log values. And you can see this here, where the percent of GFP positive cells disappears over 2 weeks, because when these don't integrate, the signal of the protein that we're delivering, GFP, is lost. The second thing that we wanted to do was to modify the envelope. And we had the Synbus virus envelope on the outside. So we designed a Synbus virus envelope, and we added it to our vector, and then we showed that this envelope is present on a Western. And we also showed that this envelope gets processed as expected. By processing, I'm referring to a furin cleavage site that's present on this envelope. And this is present on many viruses, and this allows the processing of this. And then we just had to make sure this new vector that we made, because it's a brand new novel design, still works. And this graph on the bottom right is showing that it still works. And then we had to modify this vector so that it could infect dendritic cells that express the molecule DC sign. DC sign is a receptor, and the DC sign receptor likes to have high mannose content proteins. So we had to make a viral vector that has high mannose on the outside. What am I talking about, high mannose? Well, all the sugars are processed in the endoplasmic reticulum, and as they go through the ER, there are many things that happen to the sugar moieties that are linked by asparagine on the protein. And some of these are mannose chains, and we used a drug called kifunensine. And kifunensine is added during manufacturing of these viral vectors, and this allows us to make a high mannose viral vector on the outside. And as you'll note here, as the concentration of kifunensine increases, so does the ability to infect cells. So then we did that. We made high mannose that's able to recognize the DC sign. Oh, by the way, with Western, we confirmed the presence of this high mannose on the outside of this viral vector. And then the most important part was we had to introduce a molecule called VPX. So what is VPX? Let me show you. I shared this earlier here. This is the restriction diagram I showed you earlier. Dendritic cells have an immune molecule called SAMHD1 that blocks viral infection. So we had to overcome this to make a successful gene therapy. We introduced a viral protein called VPX into this viral vector. And that's what this VPX here is. And we demonstrated the presence of VPX with a Western blot. And then we showed the ability to infect cells. So here on the left, we are adding viral vector to infect dendritic cells. And this is a flow cytometry plot. If you're not familiar with flow cytometry is a big machine that allows you to run cells through it. And if the cells are infected, it will appear a different way. If the cells are not infected, it will appear a different way. And here, in all these boxes, these dots are cells. And if they're on the left side like this, they're uninfected. But if they move into this box, they're infected. So we noticed that if we just add kifunensine alone, this viral vector doesn't work. It's not able to infect the dendritic cells. If we add VPX alone, it still doesn't work. But if we add the combination of kifunensine and VPX, now it works. And why is this? Well, because the viral vector that we're using on the outside has a mosquito virus envelope, a Synbus virus envelope. And it needs to have high mannose. So we added kifunensine to make sure it has high mannose, number one. Number two, on the inside, we needed to make sure it had VPX so it could overcome these blocks to infection inside of a cell. So only when you have both of those things were we able to perform this work. And this drug became FDA approved for clinical trials. It started its clinical trials in 2014, and we cured many patients, specifically patients who had sarcoma. So fingers crossed, hopefully this drug continues. Now, this drug was purchased by Merck because the company got acquired by the company called Merck, and hopefully it does many great things. So that's a short example of how you take something that doesn't really work and then how you do the research to make it work. And the type of research I'm showing you is a good example of research that takes place in industry because it's applied and it has a specific goal, meaning you're trying to make a therapeutic work. Okay, so then last but not least, let me tell you what happened with that as a follow-up. So when you have a drug that works like this, as I showed you in this paper, then there will be clinical trials, and then after the clinical trials, there could be an approval one day. And it's very common in companies where these products get purchased by other companies or the companies get purchased by other companies. This is very typical. But what this work led to is of course, a patent, because in the industry field, when you do research, a lot of your findings get patented. And if you were curious, the way to look up, so I showed you how to look up publications under PubMed. And if you want to look up patents, you can simply use Google Patents. And if you write a name of an inventor or anything else, then you'll see. So for example, these are my patents over here based on the discoveries I've made with my team over the past years. And if you browse through these, the discovery that I just showed you is shown right here. So it resulted in 2 separate patent filings. This is one of them. And in patents, so this was filed here in the United States, and that's why it has a US number for a patent. And here are some of the figures shown on the bottom left of what's submitted for a patent. And the patents are very long. Patents are hundreds and hundreds of pages. So for example, here, look at the scroll bar on the right. See how many, this is how long these patents are for this one particular invention here. And so anyway, I'm just sharing this to give you an idea of what the patents look like. And so often as a scientist working in industry, your life is involved in research, making discoveries, making novel findings that allow you to advance therapies. And also they allow you to make sure that you can develop drugs that will hopefully treat patients. And part of doing that requires you to work with fellow scientists, other lawyers and patent attorneys, and so on and so forth. So I think that's the end of my presentation. The last thing I wanted to mention is, as a scientist, for the last 2 years, I've been busy doing a lot of science communication. So on YouTube, I made a channel called Virus Phantom, where I talk about COVID-19 vaccines and mutations. And I also tweet a lot on Twitter, on Instagram, also under Virus Phantom. And then some funny things also happen out of this. So for example, in the United States, there are a few YouTube channels that invited me. They had me talk to children about virology and so on, and some funny things like that. But bottom line is, thank you so much for your time. I hope this was helpful. So you can find me on Virus Phantom on Twitter and on YouTube and on Instagram. And that's where I share a lot of my scientific work and COVID topics. So thanks. I'll take any questions.
Tim, may I ask something about your research specifically? What was the function of mannose in the viral DNA? Viral cell.
Good question. Yeah, so the mannose helps the virus bind to the receptor better. The receptor that we were trying to target is DC sign. DC sign is a receptor that's on dendritic cells, and DC sign tends to bind a lot of high mannose protein. So we were using the high mannose to help it bind to the receptor better.
I have a question about the after testing of these products. So after the biological improvements, how does the medical testing and peer review work in biotechnology field?
Yeah, great question. So the peer review process happens, there's a scientific peer review, which happens when you submit it for the journals for publication. When you submit it for a patent, there's a legal review done by a legal team that also includes scientists as well, because often these lawyers are scientists who then went on to get law degrees. And then when you submit these to the FDA for approval for clinical trials, you submit what's called a document that's, again, thousands of pages long, and it's called IND. IND stands for Investigational New Drug. And the same thing happened with the COVID vaccines, for example. You have to file an IND. And then the IND says that I want to test this on humans, and we believe it's safe. Here's why. Here is the data in animals and labs. Excuse me. And then the FDA, if they approve your IND, then you can start clinical studies. Clinical studies can be as short as a few months, or they can be as long as 15 years. It depends completely on the type of trial that you're running and the type of therapy you have. And in cancer, for example, in cancer studies, clinical trials take many, many years, because trying to have enough patients enroll in these studies is difficult. The reason it's difficult, you may be surprised to hear that, because you may be thinking there are a lot of cancer patients who are waiting for therapies. That's true. Guess what? There are a lot of experimental therapies as well. So these patients end up signing on to many experimental drug trials. So to get patients to sign on to your experimental drug trial takes a lot of effort and a lot of work and a lot of time. So it takes many years for that reason. In contrast, a COVID vaccine trial is very easy, because you have a pandemic. You have thousands and thousands of people dying every day. So all you have to do is just enroll people in a cohort in different parts of the world. And this is why the COVID vaccine trials took only a few months. And so depending on the cohort, so the term cohort means the demographic of people this drug is made for. It could be the age group, whether they're male or female, whether they're pregnant or not pregnant, whether they have other diseases and so on. Yeah, and so that's the process. And then there is also a quality release criteria. So every time you make a drug like this, you can't just make it in your lab. It doesn't work that way. It has to be made in an approved GMP facility. GMP stands for Good Manufacturing Practice. And in a GMP facilities are rare. For example, in Turkey, there are very, very few GMP facilities. And for that reason, it's taking a long, long time for a Turkish vaccine like Turcovac to be approved and manufactured and made. And these are some of the limitations that you can have, for example, in drug development. We work with very expensive, I'm saying very expensive, that's because they have a very high fee, but we work with contract manufacturing organizations, CMOs, that have very high fees and long waiting queues for GMP manufacturing of a drug, just so that we can start a clinical trial. So it has to be GMP manufactured, not just for the drug when it's ready to be sold, but also for the clinical trials as well. Great question, thank you.
I was wondering if there were any other reasons besides the reason you just mentioned, that doing researches and inventions are much more common in the US than it is in Turkey.
Yeah, thank you. Could you repeat the last part of your question? What's more common in the US than Turkey?
Such inventions and researches.
Yeah. Thank you. So I think this comes down to simply investments. So Turkey was a country that developed its own vaccines back in the 1950s. It was manufacturing its own vaccines, and it was actually selling vaccines to China and other countries. But then the Turkish industry steered into different directions. And for this reason, a lot of science investments decreased. So what I mean by science investments are labs that can manufacture scientific materials, and so on. And then another problem is, of course, the type of investments made in universities. Historically, it's better now, but historically wasn't very good. But I think Turkey is doing a great job of catching up. Plus, the United States is a very big country. So the Turkish population is 90 million or so, and then the US population is about 350 million. But then the US government specifically decided to invest in science back probably before the Second World War. And there were many factors that contributed to this. So today, for that reason, all those scientific investments are paying off because there is money to do the research, and there are facilities to be able to manufacture this. Unfortunately, Turkey didn't do this throughout the 70s and 80s and 90s. And for that reason, today we have a bit of a deficit, but it's getting better. So hopefully it will change.
If you have any other questions, you can also write in the chat box. And from the chat box, we have a question. How can we determine our own research topic among so many topics? How can we separate a subject that would be difficult for someone at the high school level from the simpler ones?
Yeah, very important question. So first of all of you are in high school, right? And it's okay if you don't know what you want to do, because in the education system, we're all required to know what we're supposed to do by the time we're 11 years old, and that's impossible. I mean, I'm 45 and I still don't know what I want to do, but I at least do everything I love, which is science and music and movies. And so the same thing is important for you as well. You want to do what you love doing, and don't just study something because you think it will get you a job. I know it's important to think that way, but if you really work on something that you love every day, you'll be successful. It will work out. You'll be successful because often you have to work on that thing so hard that whether you get paid or not doesn't matter. So let's say you want to become a scientist, and let's say you want to become a life scientist like me or other fellow scientists. So then you most likely want to get into research. Now, in Turkey, this might be different today, but when I was in university in Turkey in the 90s, I thought science research meant medical school. That's it. It was medical school, or you go off and become a lawyer, or you become an engineer. And I didn't realize that you don't have to go to medical school to become a researcher. In fact, it turns out that medical school is a very different education than becoming a researcher. If you want to become a researcher, you go get your PhD. A PhD in England, it's called a DPhil. In Germany, I think it's called something else. So depending on the country, it changes. But in the majority of the world, in the English-speaking world, it's a PhD. And PhD stands for a Doctor of Philosophy. And you become a doctor in a specific field of expertise. So for example, my doctorate is in molecular and cellular biology. All right. And then when you're doing this, you have to find a great school. And in Turkey, there are great schools. In Europe, there are great schools. In America, in Asia, there are great schools, right? So there are plenty of great schools. And then last but not least, you want to find a great mentor. So my recommendation to all students is when you go to the university, so the way it works for a PhD is you get a bachelor's degree, so licence diploma, right? You get a bachelor's degree first. So I got mine in microbiology. And when you get that, you get to meet the professors. And then I recommend you to find a professor that you connect with, a professor who's going to be very interested in your work, a professor who you will enjoy working with for years, because sometimes PhDs can take up to 10 years for some people. It's almost like a marriage, because you'll be working with that person every day for 10 years. So, and then after that, of course, find a topic that you want to study. I wanted to study viruses, and I wanted to specifically study how viruses evolve and mutate. So now I can use that knowledge to design viruses for gene therapy and cell therapy. And most importantly, have fun. Have fun, and remember that you will never know everything. What I mean by that is you have to be humble in this industry, because as soon as you act like you know everything, people will notice that, and you won't be somebody that they'll want to work with. So science is based on collaborations. So you want to be a good colleague, you want to be of course, good scientist, and most importantly, you want to remain humble. And unfortunately, one of the things I'm seeing right now on social media because of COVID is many professors from all over the world, from Turkey, from the United States, from Europe, docents, you name it. You're seeing people with so many egos who think they figured everything out, and you don't want to be that person. So because people will never want to work with you if you're that person, right? So many things are important, my friends, and just enjoy what you're doing. And if you enjoy what you're doing, you'll be happy either way, and you'll be successful. Also, another advice is don't be shy. I know it's easy to, I mean, we're all, you know, we all, sometimes we don't know what questions to ask. We don't know what to say, but don't be shy, because I'll give you an example from the United States. If you come here as a PhD student and you want to be successful, you have to be an entrepreneur, you know, in Turkish, girişimci, right? And you have to take charge of things. You have to talk to people. So that's another skill you should also acquire. So be comfortable talking to people, be comfortable asking questions, don't be shy. And also on that note, if you have any more questions today, don't be shy, feel free to ask.
I have one about teamwork in research, and what makes a research group a good team? What makes a team real good?
That's very important. In fact, it's so important. So at work, so our company is right now about 500 people. And in our company, we spend a lot of our time making sure the teams are the best optimal they can be. When we interview people to join our teams, we have very rigorous interviews. And those interviews allow us to select, hopefully, the best candidates so that we can hire the right people and make sure that teams continue. So what works in team dynamics is being humble and working every day to make your colleagues successful. So what I mean by that is every day I go to work and my job is to make my team successful. So I have people who report to me, I have people who I work with, and my job is to make them successful. And you know, when you think about this, you slowly realize that this isn't about you anymore. It's not about you, it's not about your research, it's about curing patients. And when you backtrack from curing patients, you realize for us to cure patients, all we have to do is make a successful drug. And to do that, we just need, like you asked, a very good operating team. And those team dynamics, when everyone is looking out for each other, when everyone is making sure the other person is successful, that's how a good team thrives. And you know, the simplest ways to say this is jealousy has no place in science. You know, people sometimes can get jealous of other projects, etc. And then especially during your PhD, you can be so focused on your own research that you may think it's always about your project, your success, getting your grants, etc. But in the real scientific world, none of this works. And again, it's another sad thing I've noticed on social media is how jealous people can be of each other's success. And so instead, we need to be happy for others' success, and we need to work to make others successful, because guess what? You'll be successful by doing that. And then in the team dynamics, there are many personalities, but usually a good lab will find all the right people to work with. It doesn't mean they all have the same personality. No, it's very good and healthy to have different personalities in the team, but those different personalities can work together. And if you don't do that, it creates a very toxic environment. And by the way, just so you know, I don't know how it is in Turkey, but in at least even in the US, you know, in many US companies, they don't do this well. But I'll say on our company, if anyone is deemed to be toxic or a toxic worker, we fire them, we let them go. So they can be the best scientist, they can be a Nobel Prize winner, they will still get fired, because teamwork is that important. And you'll see many examples of this in the news as well, where you know, for example, at our university, there was a very successful professor. He had a large lab, he had millions and dollars of grants, but he got fired He was a tenured professor, and tenured professors normally can't be fired either. But he was fired because he was toxic. He was toxic to the environment. He was toxic to his colleagues, and he's gone. And, you know, I see this on Twitter today. I don't know these people in person, but based on the way they're posting, based on the way they are demonizing other fellow scientists just for their own name, I mean, I'm amazed they are still working at the universities they're working at. So at least, you know, this isn't a comment on those universities. This is simply for you. If you are to be successful, you want to be aware of these things, because toxic attitude or toxic workplace won't be tolerated. And on the contrary, a healthy attitude will flourish, and you will be very successful, and you will be cherished. You'll be very valued by the company or the university that you work for. It was a great question. Thank you for asking.
So what drives you to research about viruses?
Yeah, that's a good question. So many reasons, but it probably goes to my childhood. And I'll tell you a few stories about that. So when I was a child, I was in Izmir, and I was living in a neighborhood called Alsancak, and we were playing on the streets, just like any kid did back in the 80s. And, you know, my mother wanted to make sure we would come home for dinner before the, you know, akşam ezanı, right? And we never did. So we were naughty. And anyway, so one of these days playing on the streets, there was a cat, and we were loving this cat, and it was a beautiful, beautiful cat. And then the cat died. And then one of the florist shops, Çiçekçi, they thought of testing the cat. They sent the cat for testing. And I'm glad they did, because it turned out the cat died from rabies, kuduz. So rabies is the deadliest virus to humans, meaning if you catch it and if you don't get treated, you're pretty much dead. So of all the viruses, so there are millions of viruses, and the deadliest to humans is rabies. So what happened is the whole neighborhood, they took all us kids to get a rabies vaccine. So at the time in Izmir, there was a rabies hospital, İzmir Devlet Kuduz Hastanesi, and we went there and we waited, and then we all got the vaccine. But it was a very scary environment. It was scary because there were many people in the waiting room with crying kids, mostly. Some were bitten by dogs, some cats. Some brought their baby because a rat apparently chewed their face or something. So anyway, so it was a very scary environment. And I love scary things because I love scary movies. And so I was very scared, but also I was fascinated. So I think that stuck with me. And then also at the same time, there was a wonderful Turkish film called Çocuklar Çiçektir, starring Tarık Akan and Necla Nazır. Look this film up. It's from the 70s. It's called Çocuklar Çiçektir, or also known as Kuduz. And it's about a village where there's a rabid dog. And then Tarık Akan and Necla Nazır both take all these children who were bit by the rabid dog, and they go to the nearest Sağlık Ocağı to get the vaccine. And the film is honestly a great commentary on Turkey in the 70s and the state of the villages in the 70s. And also it's equally a scary film. So these things stuck with me. And then last but not least, at the same time, the AIDS epidemic started when I was a child. So just like today, you're experiencing the COVID-19 epidemic starting and growing, I was experiencing the AIDS epidemic. And every day in the news, there were all these horror stories about AIDS and people dying horrifically. And so you know, as a kid, when you get exposed to these things, they stick with you for life. And I got fascinated by viruses for this reason. Later in the 90s, when the Ebola outbreaks started becoming more popular, I started following the Ebola outbreaks because I was fascinated by it. And then that's how I got into virology. And of course, today I use viruses because I know how they work and I can design them to cure cancer or design them to cure disease. So, and also when I was studying viruses, I learned that the majority of viruses are actually good. We always hear about viruses as bad, but that's only a small portion. The majority of viruses are good. They help evolution of life. And if it weren't for viruses, we wouldn't be here today because viruses helped shape the evolution of life and us too. So that's why.
We have another question. How would be a good research and experimental arrangement?
Could you repeat it, please?
How would a good research be like, and how would a good experimental arrangement be?
Got it. Thank you so much. Okay, so let's start with an experiment. When you get your PhD, which is, I keep saying get your PhD because mostly if you're going to be a researcher, you most likely will get your PhD. There are some researchers who don't get their PhD and they go to medical school and then they become a researcher afterwards. That works as well. But when you're done with medical school, what people often do is they do both. They go get their medical degree and then they do their PhD. Or you just do your PhD. So anyway, when you do your PhD, the one thing you learn is how to do experiments. You learn the experimental method. The experimental method is ingrained in your brain. You learn how to do hypothesis-driven research. You learn how to use the right controls in your experiments. And most importantly, and this is where I think a lot of colleagues fail, you learn how to criticize your own work, meaning you have to be your own peer reviewer. What does that mean? When you get a result, you can look at that result in many different ways. And you have to build the right experiment so that with all the controls and all the data, the result has only one conclusion. And hopefully, that conclusion may be disagreed with your hypothesis. And that's okay. In science, it's okay to be wrong. In fact, it's great if you're wrong because the biggest discoveries happened when people were wrong. Because they thought life worked this way, but then the experiments showed them it actually works this way. So if you were a biased scientist and you didn't have the right controls, you could misinterpret your data as if it is supporting what you're saying. But if you build the experiment the right way with all the controls, excuse me, and all the right setups and questions, then if you're wrong, it will tell you that it's wrong. And that's what you want. You want to know if your hypothesis is wrong without anyone else having to repeat the experiment. And likewise, you want to know if you're right for the same reasons. The reason for this is peer review, of course, because when you publish that work, it's going to go to peer review. And look, peer review isn't perfect. Peer review is similar to referees in soccer. You know, referees in a soccer game, in a football game, can also make mistakes, okay? But they're there because they're trying to uphold a certain standard. And same with peer review. So they're trying to uphold a standard. And in peer review, your name, that's how you build your name. If you become known for doing good quality science, who is okay with being wrong and finding new conclusions and new interpretations, you will be successful. But if you are a scientist who is all about being right and you just want to be right, then you won't be successful, because guess what? Somebody's going to be onto you very soon. And again, we see this, the best example of this is today in COVID-19, how the way the same vaccine data is being misinterpreted by people on social media. The way they're doing that is because they want to be right and they want the data to fit their thinking. And if you have that mindset, everything's gonna look the way you want it to. So that's a big problem. But guess what? The beauty of the experimental method is it's blind to those things. The experimental method will make sure, if you're trained well, that's why your professor is important, your school is important. If you are trained well, because there are many people with PhDs who probably weren't trained well, unfortunately. So if you're trained well, then you will be successful based on knowing the right experimental method and setting up the right experiments and being aware of making the right interpretations of your data and being okay with being wrong. I was wrong many times. I made many, many mistakes. So many of my hypotheses didn't work out, but guess what? Those ended up being the better papers in my research.
Yeah, thank you for joining us today. I think the questions were that. And thank you again for joining and giving the seminar again.
Thank you so much for your time. I appreciate all of you. And remember, you're all gonna be great scientists one day. You're all gonna be great artists one day. Whatever you choose to do, just make sure that you love it and enjoy it. So thank you so much for your time. Have a good day.
Okay. Thank you, Dr. Tören, once again for your speech. And now, as the chair of the program committee, I would like to talk about what makes HiSci, our event, special. Okay, so it's being presented now. So the High School Science Symposium, established in 2020, aims to give high school students an incentive to write and publish one of their first authentic papers. HiSci, a Robert College-sponsored initiative, is the first research event established exclusively by Turkish high schoolers for other Turkish high school students and for other Turkish high school students. So starting with this year, at HiSci, we also hold year-long academic events and presentations attended by respectable keynote speakers and academicians like Dr. Tören. Our recent partnership with notable scientific organizations like IEEE and Evrimağcı allows us to provide better and more frequent events for the scientific community. While taking part in our event, the participants develop their academic research, paper writing, and scientific thinking skills. Our participants are able to conduct their own research, create their own tools or computer programs, come up with their own algorithms, and elaborate on their own findings in an academic article following the HiSci paper format. And we can move to the next slide. So our participants are free to conduct a research, experiment, or develop a tool concerning math, computer science, physics, chemistry, biology, or an interdisciplinary area. Please make sure that you make your registrations and start your research in a timely manner. After your registration, you will have until April 17 to complete your papers and submit them. In order to avoid any formatting issues, please make sure that you work directly on our paper template provided as a Google document. With that being said, I'd like to state that HiSci accepts individual participants or groups of up to three people. So once the paper submission periods, actually, can we go back to the slides, please? Okay, so once the paper submission period is over, the review and the evaluation period will start, and we will grade all papers using our evaluation criteria and our rubrics. So the writers of our papers, top papers, will be informed so that they can present their research in our final symposium in June. During this symposium, we also have speeches from respectable keynote speakers, and we also hold the award ceremony there, where successful papers are recognized. last year, our awards included science-based summer school grants from notable partners like Özyeğin, Koç, and Sabancı universities. And this year, we will partner with even more organizations to provide further opportunities, including summer school grants, internships to our successful participants, so that they can foster their research and learning. So now actually we can look at our template. As previously stated, we ask you to directly work on a copy of this file so that you can avoid any formatting or font-related issues. First of all, we expect you to write a 50 to 200 word long abstract for all papers. There should be a section where you list around 5 keywords, as you see there. And that said, an important feature of our template is that we have a special numbering system. You should use the sections and subsections of your research papers. For example, let's say that you are conducting research about the virus, as Dr. Teren explained. So in section 2, you might talk about the symptoms of having that virus. You can explain the symptoms individually in more detail by dividing them into subsections like 2.1, 2.2, and so on. Then in a new section, which can be section 3, you can start talking about antibodies against that virus. And another important thing is about this template is the use of figures and tables. You should not open new sections for them. Instead, please place your figures and tables wherever they are needed. Name them as shown in our paper template, and don't forget to mention your in-text citations if you refer to any of your sources for the creation of your tables and figures. Likewise, you should also include your equations within your regular sections as you find the need to mention them. And finally, please don't forget to write your conclusion and the acknowledgements and references sections. For the references, please bear in mind that we follow the MLA 8 format. And for the in-text citations and listing of your sources, you should use our special square bracket format while referring to your sources. I also would like to state that plagiarism will not be tolerated at ISI, so please make sure that you are referencing all of your sources properly. We can move back to the slides, that's all. So moving on, you may see our key important dates for HiSci 22 in this slide. The registration period is open until April 3, 2022, and we'll accept submissions until April 17. And our evaluation process will occur afterwards, and the symposium will be held on June 4. And that said, we are very, very thankful for your interest in ISI. If you need anything else or have any further questions, please don't hesitate to reach out to us via email, and please check our website for further guides and tips about how to prepare your citations and the paper template. The link to our website can be found here, and it is hisci.robcol.k12.tr, and our email address is hisci@robcol.k12.tr. And you may also follow us on Instagram. We post a lot of useful information there. And our username for Instagram is hisci_symposium. And that's that. We are very thankful for your interest, and we can accept your questions about HiSci in general now. And also our participants on YouTube, you can also use the chat feature to write your questions or to state your questions. I think we don't have any at the moment, so you may kindly refer to our website, and also you can write an email to us whenever you have a question. That's perfectly fine. And that's that. Thank you very much, Dr. Teren, for your speech and participation. We really appreciate it. It was wonderful and very informative. So